Hi, I'm Carla Denise and this is my video of Glosary. Prokaryotes are unicellular, nucleus is absent, most eukaryotes and multicellular, nucleus is present. Microscope to see them, need warmth, moisture and nutrients divided in aerobes and anaerobes. Smaller than bacteria, depend of living host and non-living. Single cells, yeast of threads, mushrooms, need warmth, moisture, and nutrients. Same bacteria. Of genetics concerned with the transmission of hereditary characters in microorganisms. It's a process where a single cell divides twice to produce four cells containing of the origin and mode of genetic information. It's a process of dividing a cell and its nucleus into those cells, which each have their own nucleus. Of asexual reproduction is the most common form of reproduction. Is prokaryotes such as bacteria are large molecules where genetic information is stored. The chemical process that occurs within a living organism in order to maintain life are large organic compounds made of amino acids arranged in a linear chain. Are group of organic molecules that are insoluble in water but soluble in non-polar organic solvents. Are organic compounds consisting of one or more simple sugar than as monomers follow the general from two different species that are inserted into a host organism to produce new genetic combination. Particle of genetic material that along with others is arranged in a fixed order along in chromosomes. A diploid set of chromosomes in gametes or microorganisms or each cell of a multicellular organism. Genetic or genetically identical copy of a cell or an organism. A field of study concerned with the ethics implication of certain biological and medical procedures, technologies, and treatments. The Meltzer experiment used bacteria and observes how to replicate their DNA, obtain information that identifies then mechanism of DNA replication and rules out certain alternative theories. In vivo refers to when research or work is done within an entire living organism. In vitro is used to describe work that's performed outside of a living organism. Consists of a linear sequence of nucleides that are linked together by the phosphodiester bond. It's the set interaction between bases. First to the location of atoms in the three-dimensional space. Of nucleic acids is similar to that of protein and structure. The main difference between cell culture and tissue culture is that the cell culture in the laboratory process in which cells are grown under controlled conditions in vitro. Where grown of cells taken from a multicellular organism. 2 copies of original DNA molecule are produced. Each copy contains one origin strand and one newly synthesized strand. Commonly used in the lab to separate charged molecules like DNA according to size. It's a laboratory technique for the separation of a mixture. Methods as process utilizing the newly specific affinity of an antibody for its antigen to detect the distribution protein antigen in tissues or a cell. It's a scientific method used to measure how much light a chemical absorbs, measuring the intensity of light when light beams passes through the sample solution, based on the Beer-Lambert law. In conclusion, genetics is a science that studies the transmission of the hereditary characters of an organism, and its trajectory shows that its size of exponential growth, its contribution of the evolution is the species and the solutions, given the continuing problems or disease. Affected by their greatest advance despite the fact that some experiments go hand in hand with controversies at an ethical and philosophic level, such as cloning of animals. Thanks for watching this video.